Today, I do want to speak to all of you about the novelness of organoid studies and one preeminent area that we are helping researchers with, which is the counting and sizing of organoids within various growth assays following drug exposure or when focusing on toxicology of a treatment. So moving on, what are we actually going to talk about today? So to start, I will discuss in some detail what are organoids exactly and how are they grown? I will then touch on some of the pros and cons of organoids compared to other three-dimensional models being utilized, namely spheroids or non-adherent colonies. Following that, we will delve into and look at how organoids are ultimately being used as models in current research, with the focus being on looking at how organoids are being used in drug discovery and toxicology, including looking at some of the numerous assays being done. And what that'll do is that'll take us into the back half of our discussion today, where we start to look at these growth assays, which seem to be one of the most utilized assays currently being used in drug discovery and toxicology with organoids. Within these growth assays, we will then delve into manual versus automated counting and really look into the reasons why researchers are tending to stray away from manual counts where possible. Next, we will look at Oxford Optronics' platform, the GelCount, as a preeminent example of automated organoid counting and sizing, and I will actually provide a brief demonstration of the software for everyone here today. Finally, we will look at some real-world organoid examples of automated counting and sizing using this platform. So with that, let's begin. Okay, so what are organoids exactly? Well, organoids are three-dimensional, miniaturized versions of organs that are generated in vitro from pluripotent stem cells or organ-specific progenitor cells. Through self-organization and differentiation, organoids are able to develop into complex tissue-like structures that resemble the architecture and functionality of the organ they are derived from. As they are designed to mimic the structure and function of real organs, this actually makes them a valuable tool for studying organ development, disease modeling, drug screening, and even for personalized medicine. So how does one actually go about growing organoids? And of course, here I want to mention that the way I'm covering this will be overly broad, but does cover the general steps. So the first step in growing organoids is to obtain and isolate the appropriate cells from the organ of interest. This typically involves obtaining tissue samples from either human or animal sources and isolating specific cell types such as pluripotent stem cells or organ-specific progenitor cells, depending on the desired organoid type. Once these cells are isolated, they can be cultured and expanded in vitro to obtain a sufficient number of cells for organoid formation. This is typically done in culture dishes or flasks using appropriate culture media and conditions that support the growth of these specific cell type of interest. At this point, these cells can be genetically modified or manipulated to express specific gene markers depending on research objectives. Okay, after cell expansion, the cells are then transferred to a three-dimensional culture system that promotes their self-organization and differentiation into organoid structures. And this can be achieved using various methods, such as embedding the cells in an extracellular matrix or using a scaffold-based system. These three-dimensional culture systems provide the cells with the physical cues necessary for self-organization and tissue-like structure formation. Once the cells are in the three-dimensional culture system, they are allowed to differentiate into and mature into organoid structures. And this typically involves providing the cells with specific differentiation cues such as growth factors, hormones, or other signaling molecules that mimic the development cues found in vivo. These cues promote the cells to differentiate into different cell types and self-organize into tissue structures, resembling the architecture of the target organ. So maintenance and characterization. So at this point, organoids are typically cultured for an extended period to allow for the maturation and development of complex tissue structures. During this time, they are maintained in appropriate culture conditions, including optimized media, oxygen levels, and other environmental factors, which, on a side note, we can help with using our hypoxia workstation pictured here. Finally, organoids can then be characterized using various techniques, such as immunohistochemistry, gene expression analysis, functional assays, imaging methods, and really we can do all of this to confirm their organ-specific characteristics, functionality, and resemblance to the target organ. Okay, so how do organoids actually compare to what may seem to be similar models in colonies or spheroids? So in composition, colonies and spheroids are often composed of a single cell type, whereas organoids have the potential to self-organize into multiple cell types that resemble the architecture and functionality of a specific organ. Complexity, organoids are more complex structures that can recapitulate the cellular diversity, tissue architecture, and functionality of an organ, including presence of multiple cell types and tissue organization similar to the in vivo organ. In contrast, colonies and spheroids are far simpler structures that typically lack the same complexity and tissue organization. Comparing size and morphology, colonies and spheroids are often smaller in size and more uniform in shape compared to organoids. Colonies are typically small clusters of cells, that are randomly organized, while spheroids are circular aggregates of cells that can form in suspension. Organoids, on the other hand, can vary greatly in size, shape, and complexity depending on the type of organoid and culture condition used. And developmental stage, organoids are designed to mimic the development of a specific organ and can recapitulate the organogenesis process, including tissue patterning, morphogenesis, and cell differentiation. Colonies and spheroids, though, are typically not used to model organ development, but are often used for simpler applications in basic cell biology. And last, functional relevance. Organoids are considered more physiologically relevant and can better model the structure and function of organs in vivo due to their cellular complexity and tissue organization. Colonies and spheroids, while useful for certain applications, may not fully represent the in vivo tissue architecture, cell-to-cell interactions, or functionality of an organ. Okay. So we have many different organoid application areas, and researcher can use organoids to investigate the molecular and cellular processes that occur during organ development, including cell fate determination, tissue patterning, and organ morphogenesis. Organoids can be used to model diseases such as cancer, neurodegenerative disorders, and genetic diseases, allowing researchers to study disease mechanisms and test potential therapeutic interventions. In addition, organoids can be genetically engineered to carry specific mutations or modifications, allowing researchers to study the functional consequences of genetic alterations in a controlled and reproducible manner. And this is particularly valuable for investigating the role of specific genes in organ development and disease progression. Furthermore, organoids can be used to model personalized medicine, where patient-derived organoids are used to test the effectiveness of different treatment strategies for individual patients. This approach can help in tailoring treatments to the specific need of patients, improving treatment outcomes and reducing adverse effects. And finally, for the topic we are here for today, organoid use in drug screening and toxicology. So in drug screening assays, organoids are used to test the efficacy and safety of drugs in a more physiologically relevant context compared to other traditional cell methods. Organoids can be used to assess drug responses, including drug toxicity, drug metabolism, and penetration into tissues. And this can help in identifying potential drug candidates and predicting their effect on actual human organs. So, organoid researchers use a variety of assays for drug screening and toxicology studies to evaluate the effects of drugs on organoid models. And these assays can provide valuable information about drug efficacy, toxicity, metabolism, and penetration into tissues in a more physiologically relevant context. There are many commonly used assays for drug screening and toxicology in organoid research, which I do want to quickly touch on. First, histological and immunochemical assays. So these assays involve analyzing the histological and immunochemical characteristics of drug-treated organoids. They can provide information on organoid morphology, tissue organization, and cell differentiation. Immunohistochemical assays can assess changes in specific protein expression patterns, allowing researchers to study the effects of drugs on cellular markers associated with different cell types or disease-related pathways. In gene expression assays, these actually measure changes in gene expression patterns in response to drug treatment. And techniques such as quantitative real-time PCR, RNA sequencing, or gene expression profiling can be used to assess changes in gene expression levels, signaling pathways, or molecular targets in drug-treated organoids. Gene expression assays can provide insights into the mechanisms of drug action and can help identify potential drug targets or pathways. Metabolomics or proteomic assays involve characterizing changes in metabolite or protein profiles in drug-treated organoids. Metabolomics and proteomics can provide information on changes in cellular metabolism, drug metabolism, and cellular response to drugs at the molecular level. These assays can help uncover drug-induced metabolic changes, identify drug metabolites, and gain insights into drug mechanism of action. And the back three assays, we have functional assays. So these assays help evaluate the functional response of organoid to drugs. So for example, in brain organoids, functional assays can measure changes in neural activity using techniques like calcium imaging or electrophysiology. And in gut organoids, we can study nutrient uptake or barrier function. And functional assays can provide insights into drug effects on organoid functionality and help, again, identify potential drug candidates. Viability assays assess the impact of organoids on viability and cell death and can involve measuring cellular metabolic activity, assessing cell membrane integrity, or even evaluating DNA content. Viability assays can provide information on drug toxicity, as well as optimal drug concentration or exposure time for a particular organoid model. Last, we have high throughput assays. So these assays involve using automated platforms to screen for large libraries of compounds for their effects on organoids. High throughput screening assays can rapidly test the effects of a large number of drug or drug candidates on organoid models. And this allows for the efficient identification of potential drug candidates for further validation. Okay. So organoids and drug discovery, where does one actually start? So for those of you whose organoid research will have a focus on viability, functional, or high throughput assays, it's worth noting that many organoid researchers in the early days of this model are taking a basic approach that has been utilized as a gold standard in the cancer research community for over 20 years. And very simply, what they're doing is they're taking their organoids, they're treating them with different drug types or doses in assays comparable to clonogenic or tumor-forming assays, where either colonies, or in this case, organoids, will grow or not. Then, after some period of time, these researchers will numerate the number of organoids they have and compare growth or size metrics across treatments to assess treatment effectiveness. And these assays work extremely well in initial drug or toxicology screening, as the information garnered is informative on drug effectiveness. The pitfall of these assays is often found in the final step of numerating hundreds or potentially thousands of objects across tens or hundreds of plates. The overall speed, number of counts that can be accomplished, and the likelihood for bias can very much be determined by how one actually counts organoids in these assays. And while manual counting is still being used by many labs, automation in this final, often painstaking step is allowing researchers to do more. Okay, so let's compare these counting methods just a little bit here. So first, for manual counting of organoids, it can most often be summed up like this. A well-trained researcher under a bright field microscope finds an object and then determines if it's an organoid or not. If it is established to be an organoid, the researcher then needs to ascertain if that organoid meets the minimum size requirements. Again, if it does, then one is added to the running total and the researcher moves to the next object. Now, this next move, though, needs to be extremely systematic as not to risk recounting previously counted organoids, and the process is repeated for every object across every well and plate. Further, since organoids are grown in 3D space, this requires a researcher not only to be methodical across an X and Y axis, but they must also be wary that the Z axis must also be considered as not to miss a substantial number of organoids at differing levels. None of this is particularly easy to do in 3D space. Do note, this requires a highly trained individual, as it's not a particularly simple task, and overlapping objects are almost impossible to count. Regardless of a counter's efficiency, this will inevitably be an extremely time-consuming responsibility. It should also be noted that the repetitive and tedious task of counting organoids can cause both physical and mental fatigue, which can seriously affect the accuracy and reproducibility of results, especially across large counts. And studies have actually shown that human error from investigative fatigue is such a serious problem that errors of 100% or more have been observed in lengthy counts. Moreover, manual counts can be further compromised, either wittingly or unwittingly, by the bias of a researcher whose expectations about the outcome of an experiment impact their counts. The use of bright field microscopy to visually count organoids offers a major problem for researchers, which is it is often difficult to determine the acceptable organoid size across different samples. If an organoid is on the borderline of being counted, is it counted or not? Is there also potential in a particularly small set of organoids that a larger one that is under the absolute minimum value is still counted? In practice, this means an error of only plus or minus 10% in the visual estimate of organoids across samples can result in massive errors of more than 50% from plate to plate. Since size can be difficult to determine accurately, this issue can contribute to large errors when counting organoids. Finally, without any actual quantitative sizing information being available in these counts, there is the further issue that researchers cannot truly discuss growth dynamics of the organoids themselves. As many researchers undertaking counts using a microscope will attest, while they have seen differences in their organoid sizes across treatment groups, without some way to actually quantify this, this is only an observational metric and generally not publishable. So with the many issues in manual counting, what is the alternative? So the way forward. Today, we do have modern automated organoid counting and sizing systems, which offer efficient and cost-effective alternatives to manual counts. These systems can deliver a significant reduction in turnaround time of both simple day-to-day counts or high-throughput sample assays. The automated image acquisition and analysis approach to organoid counting have been validated and found to provide much higher accuracy and precision compared to manual observer counting, which reduces both intra and inter-observer variation. On top of that, these automated systems use high depth of field imaging and combine that with powerful algorithms leading to unsurpassed quality detection performance. This detection includes the resolution of things like overlapping organoids and differentiation of real organoids from debris or other artifacts. Furthermore, today's automated systems do enable operators to set defined size thresholds for counts. An example of this being exclusion of organoids based on size or shape-related parameters, as well as general object detection sensitivity. Now, what's really nice about automated systems is they do allow for repetitive plate counting of organoids without the need to stain cells. And the lack of needing to stain organoids does mean that longitudinal studies are also a possibility for those who need data at several time points. Also, technological advancements have made it possible to integrate other analysis parameters into automated organoid counting platforms. And what this really does is it provides researchers novel insights and opportunities to capture and report additional qualitative and quantitative data, which just wasn't possible with manual counts. Researchers processing organoid formation samples with automated software can now not only generate total organoid counts, but can also collect detailed size information. And here we're talking about things like organoid diameter or volume information. This ability to measure the effects of therapeutic regimes beyond absolute organoid number, but also organoid size, make it possible for research labs to extend the sensitivity of the organoid forming assay and allows researchers to obtain previously unquantifiable information related to organoid growth dynamics. Finally, one last major advantage of these automated systems is that entire experiments can become fully automated and reproducible under the exact same settings, regardless of whom the user is. Since the defined settings can be stored for a specific organoid type, that means these settings can be reutilized for similar studies, thus eliminating most human involvement and further removing the chance of bias from a study. So just to quickly summarize these points, I know that was a lot. By standardizing organoid formation analysis automation, research labs are able to improve the quality of results while reducing the time and effort needed to manually count organoids. And this is really achieved by increasing throughput, consistency, and accuracy of generated results, all while decreasing workflow demands. Okay, so what I really want to do now is I'd actually just like to run through a quick example of what an actual advanced automated counter can do, And today I'm actually going to show you our system, which is the GelCount. And what this is this is an automated colony, spheroid, and organoid analysis platform. And while we're looking at this slide, the system really does come in two separate parts. The system imager is the white box that you can see on the left-hand side, and its accompanying software component, which of course you can see on the right. So I'm going to switch over to the software now and take a 2-second break to have a drink, and then we will continue with the software demonstration. This demo now. So here is the software effectively, and what I'm going to do today is analyze an image that was previously taken by the GelCount's hardware component that you can see here. And just for your reference, the hardware component itself can image 4 separate 6-well plates of free-floating organoid spheroids or colonies in around 10 minutes. And once these settings are all set up, the software can analyze the wells in less than 2. And I will show you that today. So again, what we are looking at here is a single 6-well plate, and I can just click through all of those wells here. It's also important to note that we can zoom in and out using this functionality or with a scroll of the mouse. So I'm just going to move on to some of course, now some of the more important aspects. So to actually process an image, we actually need to tell the software the area we want to count. And to do this, we are going to apply what we call a cell mask. And so what that is that's actually this green circle that you can see here. And basically, the way the mask works is anything within that circle will be counted. Anything on the outside will, of course, not. So the mask can actually be made larger and smaller using our mask radius tool here. And also, we can move our mask with the mask moving tool here. And beyond that, we do have a few additional tools which actually allow us to apply a mask that we have set to all of the wells and dishes, which we're not going to do at this very second. So once the mask is set, the well can also be copied across, which I just showed you. And once the masks are placed correctly, they can be saved for the specific brand of plate you are using for future studies, which should really completely remove this step in the future. So I am going to just reload the mask I used earlier, just so everything is set correctly for me here. Perfect. And let's move on to some actual functionality. So next, we are going to use the optimizer tool. which gives us direct access to the fundamental algorithm and organoid processing functions. And so by clicking this, it opens up all of these tabs, which we can see here. There's 3 of them. And what I'm going to do is I'm just going to use our basic settings today, but do notice there is an advanced tab here for examples that are maybe a little bit more difficult to deal with. But again, for most examples, we can get away with the basic. And before I move on, I do want to quickly just explain how these 3 tabs here work. And really the way they are meant to work is the tabs are meant to work in a top to bottom fashion, meaning any change made to the available setting in an individual tab will have a downstream effect on the tabs below it. So we always start from our top tab and we work our way down. And again, the idea here is by optimizing and adjusting the tools and functions provided is that we'll end up with the sort of decision making by the algorithm that we feel is appropriate for our study. So with that, we will start with our pre-process tab, which is the one we're already in. And really, there isn't much to say here, but there is one function I want to show you, which is this measuring tool at the bottom. So if I turn that on, and we are actually just going to zoom in a little bit here to make this a bit easier to see. And what we can actually do is, this can be important for a researcher trying to determine what some of their larger and smaller items look like. So, for example, if we were to look at a smaller item on screen and drag across it, we can see that item might be about 106 microns. We can do another one over here. See, that one's probably closer to 67, 70 microns. And again, it's a pretty crude tool, but it can provide a researcher some crude lengths for some of their smaller and larger items. Now, this can actually be useful because it allows me to use when we get into our colony detection tab, we can actually specify a lower limit. And for this example here, I am actually going to just use 80. So what we can do is we can actually follow that same step for the upper limit as well, but we're going to go with the preset 400 microns. Okay. So let's move on. Next, I'm going to show you the edge detection shape here, which is of course, this top slider. And this is purely looking at trying to detect organoids based purely on the roundness of their edges. And really the thing to note here is if we back off the sensitivity, we can see we'll actually lose a lot of objects. So perfect. And sorry, we're just going to zoom out just a little bit more here to make this a little easier to see. So again, if we go down, we'll see that we lose a lot of what are real colonies or organoids. And if by chance we slide it too far forward, you'll see we pick up a lot of things that likely are not organoids. And really, just an FYI, to a large extent, some of the settings that, like this edge detection and the sensitivity, which I'll touch on in a second, are really meant to be used in a trial and error fashion with your organoids or colonies, just to make sure the software is able to detect what you want to delineate a colony from the background or any artifacts. I'm going to set this about here to get it into a good range. Okay. So next we're going to move to center detection. And what this tells the algorithm is what to consider a center of an organoid and what not to. And this step can really help with distinguishing overlapping objects, allowing the algorithm to separate them from one another. So similarly, again, if I move the slider too far to the left, we'll likely lose a lot of objects, and we would see the same to the right. But again, we're going to leave this one at around 65 because I know that works for this example. Okay, so last, let's move to our fine-tuning tab. Perfect. And first, we're going to look at the good edge factor, which is our first slider here. And for this, I'm actually just going to focus on a slightly different area here. Just going to zoom in a little bit. And I'm going to focus on this darker area here. And this can be useful in removing false positives of an artifact, but it can also be used to suppress non-specific detection, especially around plate edges. So that's what we're going to show here. So again, if we were to move this slider down here, what we will see is we will pick up a lot of colonies in these areas that very likely are not colonies. So again, this can be a very, very useful tool to remove some of this non-specific detection. And we'll see that that'll work about there. We can even go a tiny bit more. Perfect. All right, so the last tool that we have here, and again, I'm not going to touch on it too much today, is we have overlapping controls. And the reason why I'm not going to touch on it is for 95% of organoid examples that are growing in 3D, it's not needed. So I am going to leave that unchecked today. And that's pretty much all the settings. Again, I just want to back out to make this a little easier for everyone to see. So once we've worked through all these settings that provide the detection rules that we are happy with, we can, in a similar fashion as we did for the mask, we can actually save our settings. And that's actually at the top here. Now, I've already done that, so I'm not going to do that here, of course. But again, I'll show you that at the very, very end, how we can run through this very quickly. And the important thing to note here is, again, once these things are saved, we can come back to future counts weeks or months later and redo them. But also, we can use this for plates that use this exact same cell type and never have to reset these settings again. Okay, so once we're comfortable with our settings, we're going to click OK. And now what we can see here is that we have counted this one well extremely quickly. Also importantly, from a data perspective, is that all of our data parameters are displayed in this left column here. And so you can see things like our total counts and our diameters, along with their respective standard deviations in brackets. And so at this stage, as I mentioned, we have only processed this one well. And I just want to quickly just show you how quickly we could actually process this 6-well plate once everything is set up in the system. So what I would generally do at the start of an experiment is I would load our mask, our preset mask. We would click OK. We would then go into our optimizer tool. We would load settings. Here's my example settings from earlier. We would load those. We could click OK. Again, we're just going to recount this one well. Then we can click Recount All. And what you will notice is once it is done counting all of these, they will all turn red. So we have counted a 6-well plate in a matter of moments. And again, if we had 4 6-well plates as opposed to the one that we're looking at here, I think it's pretty easy to say that we could have counted them all in under a minute or two. Now, before I move on, there is an option at the bottom here to create a histogram to visualize some of the data parameters. So these are things like our diameters or nearest neighbor distance, and really work well as preliminary graphs to let you know what's going on. And really what I'm going to do now is I'm going to move on to our last piece of functionality with the GelCount, which is the ease of exporting data. And to do this, all you would need to do is click this save button down here. Now, just for simplicity's sake, so I don't have to bring up all these various files and click through them, I'm actually not going to click the button, but I will show you the type of data we can generate with a system like this. And I'm going to jump back to my PowerPoint to show you that. Okay. So there are really 2 data exporting options that researchers need, which are the Excel output and the publication-worthy images. So starting with Excel, we are easily able to capture all of our well-to-well items, like to our total counts, our diameter information, our average areas, along with their respective standard deviations. Now, it's also important to note, information like your saved mask and algorithm settings are also provided here. So there's no guessing weeks or months down the road which settings you used in a particular study and which to use in your follow-up experiments using the same organoid line. Now, what I think is even more important and maybe more useful to some researchers is you actually have access to the full data from each and every counted object in a respective well or dish. So here we're looking at things like, we're looking at one well from that example that we just looked at. So this is well 1. And just so you can follow along, each line of this Excel sheet is the individual information of one counted organoid. And this means that researchers like yourself actually have the ability to run multivariable statistics across treatments, comparing things like survival, volume of organoids, nearest neighbor distance, which is just something that isn't possible with the manual counting of organoids. And really what this does is it makes what would be normally a simple organoid viability study a much more dynamic experiment by looking at a range of factors and their effects. So the last item I want to cover before we look at several real-world organoid examples is that one can garner really nice images of plates with the GelCount. And here what we can see is a processed versus unprocessed image of the same plate. This, of course, can be helpful for putting together things like presentations, posters, and publications. And do note you can take out these images as a TIFF, a JPEG, and a bitmap format. Beyond that, it also demonstrates to others in your field or your lab even that you are appropriately counting your organoids and can act as a nice check and balance to anyone's research. Okay, so now we're going to look at some direct organoid examples that have been sent to us. The groups who have sent us these have allowed us to use these images, but in many cases have asked us to remove identifying information like organoid of interest that they were looking at. Although really the main goal here, more than anything, is just to cover several different types of organoid examples we have seen in the past and demonstrate how the GelCount handles these. So starting with this first image, we can see an unprocessed image, an analysis image, and then a fully counted image in which it looks like all the organoids are indeed counted. Next, here is an example of a larger extracellular matrix dome filled with growing organoids. Again, for this study, you'll notice that they have 24 well plates of these domes. And once the algorithm was set up, we were able to count all 24 wells consistently in a minute or two, whereas manual counts would take probably hours for these hundreds of objects in each well. All right, so this is actually another dome example, but this time it's multiple domes in a 6-well plate. And as we can see, these are bladder organoids. And for ease here, we have used individual red dots to capture the objects rather than the circles to make it far easier for us to just kind of see these counts. But as you can see, there is a ton of objects in each one of these wells, or each one of these domes, sorry. So this is a fairly simple example, but I like this one because what it demonstrates is the GelCount's ability to resolve overlapping in close objects. And again, if you look at the processed versus unprocessed image here, it looks like we are doing a fairly good job of resolving what look to be very, very close objects with our counts. All right, the next one here, this is a bit of a different example, but is meant to demonstrate that there's an option to resolve larger and smaller organoids independently of one another. So to do this, we do a dual run mode, which makes this a possibility. And you can see the top image counts the larger objects, while the lower image focuses on the smaller ones. Right? And the last two examples that we have here are both pancreas organoids from the same sample and show what I tend to call later stage, almost bubble-like organoids. And really my opinion here on these two is we're actually able to get very decent counts, but I would tend to say this is a little bit unreliable as it's very clear that the organoids grown here are growing very much on top of each other and are far too concentrated with objects to get a really reliable count. And again, here's the second image. And our suggestion, while we can count this, would be that these images perhaps should be taken when the organoids are smaller, or if they need to be seeded to this rate, that they do be perhaps maybe either seeded a bit less or the domes are made a little bit larger. But again, you can see how we are able to get counts here where somebody manually very likely would not be able to. And the last example here is one taken from the one of the first papers to use our system with organoids in 2020. And as a side note, since this paper has come out, we are seeing papers involving organoids and the GelCount far more frequently month after month, as this novel model is used more and researchers are seeing they can automate the counting and sizing of organoids while removing bias. So back to the example, Chakraborty and team developed a CRISPR-edited organoid line, and without getting too deep into the study, Here, they use the GelCount to provide both the well image, as you can see, of the control and the BRCA2 lines, but they also did their analysis, which clearly showed a higher number of organoids in the edited cell line over the control. And one final point before we close out our talk today, I do think I've supplied enough information on the GelCount, but I did want to mention for researchers who want to simulate the in vivo environment more closely for their organoid lines, I would very much suggest looking at our hypoxia lab as well. This is a system that allows one to precisely grow and maintain organoid culture under oxygen levels that are far more relevant than the 18 to 21% inside a standard CO2 incubator. And just for your information, this benchtop system can control oxygen, carbon dioxide, temperature, and humidity all on that front-facing touchscreen. So I'll just say certainly do keep that in mind as well.